This is podcast number 42. Your final is on Monday the 7th from 12:15 to 2:15. Make sure you get that right. Normally we have our tests on Fridays, but this is finals week, so your final is on Monday, and it's also at a different time. It's going to be online like you've taken your other tests online. This one counts for 25% of the course grade. It'll have 75 multiple choice questions. There are 43 podcasts, but 4 of those are review. So let's say 39 podcasts plus 13 recitations. So about 1.5 per podcast and recitation. 1.5 questions per podcast and recitation. Those are worth 2.5 points apiece. You'll have 9 extended response questions, and you have to choose 5 of those to answer. They'll be worth 12.5 points apiece for 250 points total. And because something is in this review or it's not in this review doesn't mean anything. This is just to jog your memory. Anything that we've talked about in this course is fair game for the final. I ask you to draw a mitochondrion, and I likely will. Then I want you to draw a very simple diagram. So one line is one membrane. The outer membrane is the one I just drew. The inner membrane is the one with the cristae that is folded up. So this is outer. Inner. This is the matrix all the way in this space here. Matrix. And this is the intermembrane space. Inter with a T, not inner with an N. Inter, it's between the two membranes. I also might ask you where things are happening. Protons moving during electron transport. Where are they moving during ATP synthesis? Where is pyruvate moving in stage 2? That sort of thing. Okay, let's now, if I would, chloroplasts have 3 membranes. 1, 2, and then the third one is the thylakoid membrane. That's the innermost membrane. Again, 1 line per membrane. Okay, so these are the outer. And this is the thylakoid membrane. Stack like this would be, or several stacks like this would be grana. Grana is plural of granum. And then the space outside the membrane is called the stroma. Space inside the membrane is called the lumen. And again, I might ask you, where does the Calvin cycle happen? Where do the light reactions happen? Where are protons pumped? That sort of thing. Picking up our review from podcast number 42, we talked about different metabolic pathways, including beta oxidation. We talked about how beta oxidation cuts at the beta carbon in the acyl chain tail of fatty acids, and then ultimately produces an acetyl-CoA, which will enter the TCA cycle. We talked about how the TCA cycle is sort of a crossroads for lots of different metabolic pathways, whether you're breaking down carbohydrate or fatty acid or protein. Here's an H5P question for you. We talked about the structure of nucleic acids and how that structure can affect whether or not you are able to access that DNA for transcription. So your DNA is wrapped up in histones, which are proteins that organize your DNA. Histones are arranged in different subunits like so, and there is a certain amount of DNA that is wrapped around each histone, 146 base pairs of DNA, and then there's linker DNA between them. Those histones are part of bigger structures, which fold into bigger structures and eventually fold into chromosomes. How the DNA is wrapped can be modified by chemical modification, either attaching to the DNA or attaching to the histones. A chemical modification, common chemical modification to the DNA called methylation is adding a methyl group onto the histones, and that leads to tighter packing of the DNA being less available for transcription. And then acetylation, another kind of modification of the histone itself, leads to looser packing of the DNA, making it more available for transcription. These are modifications that happen during an individual's life and can affect their ability to transcribe genes. So without mutating the DNA or changing the sequence of the DNA, you can change the access to the DNA, and that can have an effect on the individual's phenotype. We talked about how DNA is replicated in multiple origins of replication. In the case of eukaryotes, a single origin of replication. In the case of prokaryotes, we talked about how in other classes you were introduced to replication forks, and replication forks is just half a replication bubble. And in the bubble, it's very important to know which direction the bubble is opening. So on this side of the diagram, it's opening off to the left. On this side, it's opening off to the right. And that will change what is the leading or lagging strand. So when you are indicating what the leading or lagging strand is, make sure that you are pointing to the correct strand. So that's the newly synthesized strand is either leading or lagging, not the template strand in the darker blue here. Notice the arrow goes to this and to this, not to that. Okay, so you want to be very precise when you are drawing a diagram like that. We talked about how there are thousands of replication bubbles all opening at the same time when a chromosome is replicating. Otherwise, it would just take much too long to duplicate. Here's an H5P question for you. We talked about lots of different mechanisms of DNA repair. This is one called synthesis-dependent strand annealing that's only available when your chromosomes have sister chromatids, because it uses the sister chromatid as a template to repair a double-stranded break, to see what the nucleotides were in the part that's missing, and then synthesize those according to what the sister chromatid has. But that's one of many kinds of DNA repair mechanisms. Here's another H5P question for you.